In the final lecture of the chapter on carbohydrates, we will look into some of the oligo and polysaccharides. So we'll look at a few examples and look at their properties and maybe some reactions. In a disaccharide, two monosaccharides form an acetal bond. Reverse reaction is also possible, and disaccharide can be hydrolyzed into its constituent monosaccharides. Some of the common disaccharides are sucrose, lactose, maltose, and cellulose. We will examine these four, and then we'll move on to oligosaccharides. A disaccharide can be homodisaccharide, that means it's composed of two identical monosaccharide units, or heterodisaccharide, and that one is composed of two different monosaccharide units. An acetal bond that joins two monosaccharides in a disaccharide is called O-glycosidic bond or oxygen glycosidic bond. And here we have an example of sucrose. So there is O-glycosidic bond between monosaccharides glucose and fructose. Glucose is shown in red and fructose in blue. As you can see, this is a heterodisaccharide. So two different monosaccharides bonded to form a disaccharide. On the left is formula composed of two Hayward projections. On the right is formula that is more commonly used in biochemistry. And note the angled bonds that glycosidic oxygen forms. So bonds at right angle. That's one bond. Unlike organic chemistry, where each such bond would actually represent CH2 carbon, and would really mean that there is a carbon between oxygen and carbon 1 of the ring. In this case, it simply means that bond points down. So this is a biochemical convention to clearly show whether the glycosidic bond is alpha or beta. So we draw a bond straight down or straight up, and then continue that same bond at 90 degrees to form the complete bond to oxygen. So part of the bond, half of the bond that points straight down or straight up, indicates whether bond is alpha or beta. And then horizontal part of the bond continues that bond to form a bond to oxygen. From the formula, you can probably draw a conclusion that sucrose is not a reducing sugar because it doesn't have a free hemiacetal group. Actually, it's two hemiacetal groups, two hemiacetal hydroxyl groups that react to form the glycosidic bond. So both glucose and fructose are in the form of their respective acetals in the molecule of sucrose. Sucrose can be hydrolyzed, and hydrolysis can be carried out either by enzyme sucrase, or it can be hydrolyzed by treatment with a dilute acid, such as hydrochloric acid. And of course, it's hydrolyzed into glucose and fructose. Glucose and fructose are, of course, reducing sugars, and so hydrolyzed solution is reducing solution. Maltose is a product of hydrolysis of starch. It's a disaccharide, and it's homodisaccharide because it's composed of two units of glucose. They are joined by alpha 1,4-glycosidic bonds. Bond is indicated as alpha glycosidic bond based on the stereochemistry of bond on carbon 1, because that's the anomeric carbon, and that glycosidic bond could be alpha or beta. If monosaccharide is glucose, as in this case, then on carbon 4, bond to oxygen has to be alpha, because in glucose that bond is always alpha. So if we use term alpha or beta to refer to glycosidic bond, that refers to stereochemistry of bond on carbon 1. Maltose can be hydrolyzed to glucose by enzyme maltase. Cellulose is related carbohydrate. It can be obtained by enzymatic or acidic hydrolysis of cellulose. And it's also homodisaccharide. When it's hydrolyzed, it of course gives glucose. And unlike maltose, which has alpha 1,4 glycosidic bonds, cellulose has beta 1,4 glycosidic bonds. Another interesting disaccharide is trehalose. It's naturally occurring homodisaccharide, so it's composed of two identical molecules of monosaccharide, and again monosaccharide is glucose. But now two molecules of glucose are linked by alpha 1,1 glycosidic bonds. That means both glycosidic bonds are alpha, on both units of glucose. And it is non-reducing sugar because there is no free hemiacetal bond. So this, unlike previous two sugars, maltose and cellulose, which were reducing sugars, this one is non-reducing. It can be hydrolyzed to glucose by the enzyme Trehalase, and humans have that enzyme. So that means that it is energetically equivalent to glucose. It is much less sweet compared to glucose, and relatively recently, an inexpensive method to prepare it synthetically has been discovered. So it has been used as food additive because it has very high water retention capacity and readily forms gel with water. So it's used in preparation of ice cream. It makes ice cream taste really nice, smooth, and creamy. But problem is that even though it is naturally occurring disaccharide, it has been associated with sharp increase in gastrointestinal diseases caused by strain of bacterium Clostridium difficile. So it's basically that bacterium feeds on trehalose and really thrives in high trehalose environment. And finally, another important disaccharide is lactose. Lactose is also called milk sugar because, of course, it's main constituent of milk, and it's composed of molecules of galactose and glucose. Molecules of galactose, so it's heterodisaccharide. Molecules of galactose and glucose are joined by beta 1,4 glycosidic bond. It is, of course, reducing sugar. You can see that unit on the right in this formula is glucose unit, and that one has free hemiacetal, which means it's reducing sugar. In humans, lactose is hydrolyzed by the enzyme lactase. Another interesting disaccharide is heterodisaccharide lactulose. This disaccharide is composed of galactose and fructose joined by beta 1,4 glycosidic bond. It is a reducing sugar because fructose unit has free chemical function. Lactulose is not present in raw milk, naturally occurring milk, but it is produced upon heating of lactose. So lactose, when heated, when milk is, for example, pasteurized, will partially, to small extent, be isomerized into lactulose. Humans and quite a number of animals cannot digest lactulose. So it has medical applications, or in case of animals, veterinary applications. It can be used to test for the presence of certain bacteria in the gut, because bacteria can digest it and then can produce hydrogen and methane, which can be detected in one's breath. And also it is used as a laxative, so-called osmotic laxative, because of the large number, it cannot be digested and has a large number of hydroxyl groups, so it absorbs a lot of water, so it swells as it passes through gastrointestinal system, and that way it serves as a laxative. It is also a laxative in veterinary medicine. So, for example, if your pet is constipated, lactulose may be prescribed. But I can tell you, good luck trying to give it to a cat. And we will look at raffinose as an example of a trisaccharide. Raffinose is a heterotrisaccharide. It's composed of monosaccharides galactose, glucose, and fructose. They are shown here. Individual units are shown here galactose in green, glucose in red, and fructose in blue. It is non-reducing sugar. You can see that there are no free hemiacetals or hemiketals, so it is non-reducing, and it can be found in various vegetables such as beans, cabbage, Brussels sprouts, broccoli, asparagus, whole grain. It is hydrolyzed by D-galactose and then remaining sucrose by alpha-galactosidase. Alpha-galactosidase is not present in human digestive tract. So Raffinose passes through the stomach and upper intestine, and then in the lower intestine it is fermented by gas-producing bacteria that do possess alpha-galactosidase. And they produce carbon dioxide, and so they methane and hydrogen, and so that leads to flatulence that is commonly associated with eating beans. So various digestive aids, such as Beano, contain alpha-galactosidase. Polysaccharides are polymers that contain monosaccharide units as monomers. They may be homopolymers if monosaccharide unit is the same, or heteropolymers if they have different monosaccharide units. We will examine only several homopolymers, and they are going to be starch and glycogen, cellulose, and chitin. So first we'll look at starch and glycogen. Starch is used as energy storage. It's a nutritional reservoir of carbohydrates in plants. And there are two forms of starch, amylose and amylopectin. Both of them are hydrolyzed by alpha amylase, and amylose is linear polymer. It contains only alpha-1,4-glycosidic bonds. Amylopectin is branched. So in addition to alpha-1,4-glycosidic bonds, it also contains branching points that are alpha-1,6-glycosidic bonds. And there is one branching point approximately every 30 glucose units. In mammals, carbohydrate energy is stored in form of glycogen. Glycogen is the most common copolymer in animal cells, and that's actually how glucose is formed. It resembles amylopectin, but it's far more branched. Branching allows for faster hydrolysis because hydrolysis occurs at the end of a chain. And of course, by being branched, it has many more branches and therefore more ends to the chain, so it can be faster hydrolyzed to individual glucose molecules in case energy is needed. Both starch and glycogen form hollow alpha helices as they coil. They coil as a result of alpha-1,4-glycosidic bonding. So the way it was shown as long chains on the previous page was not correct. Really, they are coiled like this into alpha helices, and then as a result they occupy a relatively small volume. So a large number of individual glucose molecules can be stored in a small volume, and that results in rather efficient storage of large amount of energy in small volume. As mentioned already, alpha amylase can cleave 1,4-glycosidic bonds, but that leaves 1,6-glycosidic bonds, and they cannot be cleaved by amylase. They are cleaved by alpha-1,6-glucosidase, and we'll go into more detail about metabolism of these polysaccharides in the next chapter. Cellulose, on the other hand, is a structural, not a nutritional carbohydrate. It is also, of course, homopolysaccharide and homopolysaccharide of glucose, but it has beta 1,4-glycosidic bonds. So it's beta 1,4 linear polymer of glucose. It is main constituent of plant cell walls. Humans and other mammals cannot hydrolyze cellulose, and so it cannot be used as food, as energy source in humans and mammals. But some bacteria are capable of hydrolyzing it, and animals that contain such bacteria in their digestive tracts, and those would be ruminant animals such as cows or insects such as termites, they can utilize cellulose as food. Another important structural carbohydrate is chitin. It has a structure that is very similar to that of cellulose, and also it has beta 1,4 glycosidic bonds, but carbon 2 is in the form of an amide. So on carbon 2, instead of hydroxyl group, there is 2-acetamido group. So monomer is 2-acetamido 2-deoxyglucose. And as a result, intermolecular interactions between individual chitin chain are very strong, so chitin is very strong, very rigid structural material in animals. It forms exoskeleton of insects and crustaceans. Enzymes that are involved in synthesis of polysaccharides are glycosyl transferases. Those enzymes catalyze the formation of glycosidic bonds. And each enzyme is specific to the two sugars that are being linked, so the two monosaccharide units that are being linked to each other to form a glycoside bond. So in this case, specificity is achieved through a variety of enzymes. On the other hand, in synthesis of polypeptides and nucleotides, which we'll cover later in the course, specificity is achieved by transfer from a template, while catalytic apparatus is identical in each case. So it's a single catalytic apparatus that operates. Monosaccharides that are going to be incorporated into polysaccharides are in the form of activated sugar nucleotides. And it's sugar activated by UDP, uridine diphosphate group. So glucose that will be incorporated into a polysaccharide like cellulose or amylose or something like that will be in form of UDP glucose. And UDP group is the leaving group. A biologically very important class of polysaccharides are glucosaminoglycans. Glucosaminoglycans are anionic polysaccharide chains, and they are composed of repeating disaccharide units. Those disaccharide units are composed of, each disaccharide unit is composed of two different monosaccharides, which means that glucosaminoglycans are heteropolymers. Glucosaminoglycans are classified as proteoglycans. It means that they have protein core to which large polysaccharide chains are attached. By weight, they are typically composed of about 95% of carbohydrate and about 5% of protein. So they are mainly carbohydrates. And their properties are entirely due to carbohydrate component, due to proteoglycan component, or glucosaminoglycan component of proteoglycan. They have a number of biological roles. One important role is that they are constituents of joints, so they serve as joint lubricants, structural components of connective tissues, and also as shock absorbers. You can see their structure. They are composed of not only of repeating disaccharide units, but they contain derivative of amino sugar, glucosamine or galactosamine, and at least one sugar contains a negatively charged carboxylate or sulfate group. As such, they form bonds to a large number of water molecules. So they are heavily hydrated. And since they are heavily hydrated, they are really in form of a gel. So each chain is associated with forms intermolecular interactions with large number of water molecules to form gel-like structure, and then upon shock, that those chains are squeezed and water is squeezed out. Let's say if one jumps, so that's how joints absorb the shock, because individual chains are squeezed, water in between them is squeezed out, and then as pressure is gone, they resume their ordinary shape and water flows back in, and so that way they absorb a shock. They play other biological roles. For example, they mediate adhesion of cells to extracellular matrix and bind factors that stimulate cell proliferation. Old name for proteoglycans is mucopolysaccharides, and mucopolysaccharidosis is a name given to a collection of diseases that are a result of the inability to degrade glucosaminoglycans. And some common disaccharide units that are encountered in glucosaminoglycans are given here. So they are, for example, chondroitin 6-sulfate, hyaluronate, dermatan sulfate, keratan sulfate, and heparin. Glycoproteins have names similar to proteoglycans, so they are very easily confused one with the other, but they are completely different from proteoglycans. Proteoglycans are polysaccharides, but glycoproteins are actually not polysaccharides. They are mainly proteins, so their core is a protein, but not only that core is a protein, most of the molecule, of the large biomolecule, is a protein. Carbohydrates are oligosaccharides that are attached to its surface. They are components of cell membranes, and there they play a role in cell recognition. Some of them also could be soluble proteins. Carbohydrate is attached to protein core by carbohydrate-protein link, and that link could be N or O glycosidic bond. So it could be nitrogen or oxygen glycosidic bond. If it's N linkage, then carbohydrate in glycoprotein is attached to the amide nitrogen of asparagines, provided asparagine is part of asparagine something labeled here as X, so asparagine, some amino acid residue, serine, or asparagine something threonine sequence. So two amino acids that are needed here besides asparagine are amino acids that have hydroxyl group inside chain. X could be any amino acid except proline. A potential glycosylation site can be identified from amino acid sequence, but not all potential sites are glycosylated. All N-linked oligosaccharides have a common pentasaccharide core composed of 3 mannose and 2 N-acetyl glucosamine residues, and then other monosaccharide units attached to that core. In case of O linkage is to the side chain or of serine or threonine, and that's shown here. So oligosaccharide unit shown here, only part of oligosaccharide unit is shown here, but oligosaccharide unit attaches either to part of hydroxyl group of serine in first 3 examples, or hydroxyl group of threonine in the final example. And finally, lectins are proteins that bind specific carbohydrate structures on opposing cell surface. Their main function is to facilitate cell-to-cell contact. And so lectin usually contains 2 or more binding sites for carbohydrate units. And the binding sites on the surface of one cell interact with carbohydrates on the surface of another cell. Linking is achieved through intermolecular interactions. Intermolecular interactions are supposed to be weak, relatively weak intermolecular interactions, weaker than covalent, but actually ion-dipole interactions could be pretty strong. And these interactions ensure specificity, but also while they are pretty strong, they are not strong enough to prevent unlinking. They are just strong enough to keep these units attached to each other, but then unlinking can also occur. So they are weaker than covalent bonds. Lectins are classified according to their amino acid sequences and biochemical properties. One large class are C-type lectins, that stands for calcium requiring lectins, and that's shown here. They are found in animals. Calcium ion acts as a bridge between the protein and carbohydrate unit, and it acts as a bridge through ion-dipole interactions. Lectins are examples of carbohydrates that serve as information carriers, information-rich molecules. So that's one example of the role of carbohydrates. They serve as information carriers in recognition of carbohydrates on cell surfaces. Structural diversity in general, not only in case of lectins, but structural diversity in general and relatively high information content of carbohydrates is due to a diversity of monosaccharides. There is a very large number of different monosaccharides. The fact that bonding can occur on any through several OH groups of a monosaccharide, bonding on anomeric carbon, carbon C1 could be alpha or beta, and branching of oligosaccharides is possible. So this is in contrast to other information carrier molecules, such as proteins and nucleic acids, where information is contained in the sequence of monomers and not in the diversity or mode of the bonding. So in proteins and nucleic acids, bonding is always the same and there are relatively few monomers. So completely different from the way carbohydrates carry information. And this completes our chapter on carbohydrates.